Hi guys, welcome to Cosmic Solutions, Dr. Priyanka's Biology, where we discuss everything from biology to biotechnology. So today let's get started with yet another chapter of your NCERT 12th standard textbook, chapter 11, that's biotechnology, principles and processes. So this being one very interesting topic. Okay, so here we will try to understand what is biotechnology, what are its governing principles, and what are the processes involved over here. Okay, why a separate branch of biotechnology has been established. What is its usefulness? So today let's try to decode what are the principles of biotechnology. So before we move on, let's try to understand what is meant by biotechnology. Okay. So, if we break the word that is bio and technology, that means biology. So, definitely all the streams of natural sciences will come over here. And next part is technology, the engineering science. Okay. So, basically, your biotechnology is the meeting point or the amalgamation of natural sciences and engineering technologies, okay, which are being put up together in order to gain more products or gain more information, more usefulness from the biology, okay, which is or all these things can be useful to the human race. Okay, that is what is biotechnology if we try to understand it in simple words. Okay, so let's try to understand what are the principles of biotechnology. Okay, so there are two basic principles which govern the field of biotechnology. The first one is genetic engineering. Okay, so we know we try to engineer something, okay, at the genetic level. So when we talk about genetic level, we talk about the DNA, RNA of an organism. Okay, so genetic engineering is a technique, okay, which allows us to make alterations or changes either by introducing something new or by deleting something from the original makeup of the DNA or RNA of an organism. Okay, and these changes are such a way that it causes the changes in the phenotype of an organism when it is being introduced into the host. Okay, and next part is the bioprocess engineering. So genetic engineering talks about the alterations, the changes that we deliberately make in the genetic material of an organism. Whereas bioprocess engineering, okay, so we know bioprocess, so biological processes. So here we try to provide, okay, an environment, strictly sterile environment, which will allow the growth of desirable microorganisms. So maybe we can talk about a fermenter. If we want to produce, okay, say a protein, okay, so then we need to grow that organism, that bacterium, okay, which is going to produce that protein. But this is not enough when we are just allowing one or two bacteria to grow. We need millions and billions of bacteria. That too, it has to happen within a short span of time. So, the environment that is provided for the maintenance and growth of such desirable microorganism, the processes involved in it, the processes which are involved in the production and for the purification of that particular product obtained from the organism. All these things will come under the bioprocess engineering. So which may include your downstreaming processes also. Okay. So now let's go into little more details. Okay, so now we have been talking about the sexual reproduction and asexual reproduction. So sexual reproduction, we know there is going to be contribution of both the parents. 50% will come from a mother, 50% will come from a father. Okay, this will lead to a different sort of combination. Okay, but in case of your asexual reproduction, the genetic information is preserved. Okay, so there is no alteration in the genetic makeup. Alright, so that is the natural process what occurs over here. Now when we talk about genetic engineering, okay, so genetic engineering basically it involves very specific work. Okay, for example, let's take one example. You have alphabets from A to Z, right? A to Z. There are different alphabets involved over here. And suppose if you want to select only the alphabet in the middle, say J, K and H, okay? If you want to choose only these alphabets, but you have the entire stretch of the alphabet starting from A to Z. But your job is to pick only J, K, H. So what will you do? We will have to cut and erase the alphabets before J and alphabets after H, so that we are left only with this, right? So what are we doing? We are trying to, you know, make changes in the sequence of alphabet. Okay, something similar happens when we talk about your genetic engineering. So why I gave you the example of this selecting only this part, because I want to correlate this with the specificness. Okay, same thing we are going to talk in terms of your DNA. Okay. Alright. So this genetic engineering, it includes creation of recombinant DNA. We will see what is recombinant in a minute. Then after that, use of gene cloning. Okay. And third part is gene transfer. So first thing is going to be recombinant DNA. We are talking about combination of two different DNA. So we will have one DNA, okay, and we will have another piece of DNA. So when both this DNA are joined together by some method, okay, we'll see what is that method later on. Okay, this new product is called as the recombinant DNA. Next part is gene cloning. Okay, cloning meaning identical copies but in multiple numbers, right? So when we are going to increase the number of this, okay, all right, okay, so we have this one copy of red and blue. So now what I have done is I have increased the number of these red and blue. So here what will happen? Okay, it has multiplied. Okay, this particular thing, the recombinant DNA has multiplied, that is increase in the number of copies. So what has happened? It has caused cloning. Identical copies have been produced. So this is called as gene cloning. And next part is gene transfer, is when we will introduce these genes, okay, into some host organism. So okay, so the, okay, when we introduce these copies into a host organism, it is called, that method is called as the gene transfer. So whatever I have explained you so far is using no technical terms. It is just for your understanding. But now in coming slides, we will talk more about the technical terms. Okay. Now, what has happened over here is only the required set of gene has been transferred over here. Okay, suppose now, alright, this original DNA, pieces of DNA had this yellow part also, but what we have done is we have removed off this yellow part. Yes, we have removed off this yellow part, and allowed only this red and blue part to be joined, right? So what has happened? It is specific. Only the desired genes have been introduced. Okay. Alright. So let us see some more terms. Now let's talk about something called as plasmid. Okay. So plasmid is basically used as a cloning vehicle. Okay. Now um, suppose now if you have to travel from place A to place B, okay, what will you do? You have different modes of transport, right? For example, let us take a public transport, that is your bus. Okay, so this plasmid is like a bus. What it will do, it will take some passengers, okay, it will make them sit, it will make them settle, and it will take them to a different location. Okay, so when they reach the location, then again the passengers are free to either move out or sit inside the bus. So same thing is done by your plasmid. So plasmid is, okay, if we have to define plasmid, it is a extra chromosomal DNA. Suppose now we have this bacterium, okay, it will have its own genetic material here called its own DNA, and your plasmid will be something that is something other than the genetic material. Okay, so it is called as extra chromosomal. So it is not a part of the chromosome of your bacterium. So it is the extra chromosomal DNA which is capable of self-replication. So we know that your genetic material undergoes replication, right? So it will have its own machinery, and your plasmid will have its own machinery to self-replicate. So this is what is called as your plasmid. So I told you this plasmid is going to act like a bus which will carry the passengers. So suppose now we have yet another bacterium, its own genetic material here, and here we have, if I want to introduce some gene, okay, for example, now I have done some changes and entered some pink color gene in this plasmid. So what will happen? We can introduce this entire plasmid into your new DNA. Sorry, into a new bacterium. So this is called as transformation. We'll talk about it later. So what I'm trying to explain you is the why we can call it as a vehicle. So this plasmid is going to allow, or it will take this piece of DNA into the new bacterium. Okay. So that is the role of plasmid. So this plasmid were first constructed by Stanley Cohen and Herbert Boyer. These two people, they constructed the first plasmid in the year 1972. So what they did was they isolated, okay, the antibiotic resistance gene. Okay, so a gene which is responsible for causing resistance to antibiotic. So they isolated that gene from some bacterium, okay, and what they did, they cut it, okay, and they took the piece of that antibiotic resistant gene, okay. They saw that this new piece of DNA was able to confer the antibiotic resistance. Okay. So this was first studied in Salmonella typhimurium. So the organism which is responsible for causing typhoid. Okay. Now they use something to cut the DNA. So what did they use? Can they use scissors? No. But they use something called as the molecular scissors. A molecule which is responsible for making cuts or nicks. More technical terms is nicks into the DNA. So they are called as the molecular scissors in general, and the technical term is restriction enzymes. So basically these molecular scissors are the enzymes which have the property of cutting. So cutting is basically breaking the bonds in the DNA. Okay. Now, suppose we have the plasmid over here and a piece of DNA. Okay, we want this piece of DNA. It is coding for something. Say antibiotic resistance. Okay, it is coding for some antibiotic resistance. So I want, okay, suppose now, okay, this from this black part, only this white part is responsible for antibiotic resistance. Okay, so I don't want this black part to be inserted in this. Okay, now we have to look for the area where this restriction enzymes can cut. Now what scissors we have, we can cut anything that we want. Okay, now suppose let's take an example. You have a very small scissor. Okay, suppose if you have a very small scissor, and that scissor we can use only for cutting thin sections, okay, say some paper, but we will not be able to cut a thick piece of cloth or thick piece of something with the help of that small scissor, because it is made in such a way that it is going to cut only a small part. Okay, otherwise let's take yet another example. Okay, suppose now I have this piece of paper. Okay, a piece of paper is there with me and I have this notepad. Okay, so the thickness of notepad is slightly more as compared to the thickness of this piece of paper. Suppose now, okay, I join both the things. Okay, I join both the things and I want you to cut this together. But the scissor that I have is very tiny, very small. So it will be able to cut only the area in this small paper and not the area of this particular notepad. Or forget notepad, because you may say that it is of same thickness. Okay, this notebook and this piece of paper. So there is a difference in the thickness, right? So we will have to use a bigger or a thicker scissor to more sharper scissor to cut the book rather than the paper. Paper can be cut with a small scissor. So why I am giving you this example is basically I want to tell you that this restriction enzymes are also designed in a very specific manner, that the way I need a particular type of scissor to cut this piece of paper and a different scissor for cutting a thicker part of that book. Okay, same way different restriction enzymes are required for cutting different types of DNA. Okay, we simply cannot use any sort of DNA, restriction enzyme for cutting any type of DNA. That is point number one. Point number two is that they have specific area. If I have a big piece of DNA, it is not necessary for a specific restriction enzyme to cut each and every part of that DNA. But it is dependent on the sequence present on that DNA. It has to recognize a particular section, a particular sequence, okay, and then only it is going to cut or produce a cut, okay. So that is the specificity of your restriction enzyme. So the role of restriction enzyme is to cut the DNA. But what do we have to join? We need some glue. Okay, and that glue is called as the enzyme ligase. Okay, it is enzyme ligase. Ligation is the process of joining. So restriction enzyme are the molecular scissors which will cut the DNA in a specific sequence, whereas the ligase is the glue which is going to join two strands of DNA or two pieces of DNA. But the correlation between your restriction enzyme and the ligase is that the piece of DNA, okay, has to be cut. Okay, if we have two pieces of DNA, we are taking this example, then this has to be, this plasmid DNA also has to be cut by the same restriction enzyme, and here also this piece which is having the antibiotic resistance has to be cut with the same restriction enzyme. Then only these both the pieces can be joined together by your ligase enzyme. Okay, here your ligase enzyme, okay, it is going to join the piece which is having the antibiotic resistance and the other part of the plasmid. So this is the role of your restriction enzyme and ligase. So these two form the most important tools in your genetic engineering. All right, so let's move on to the next part. All right, so far we have discussed about restriction enzyme and the ligase. Okay. Now let us talk about recombinant DNA. So far we had spoken about it in layman's terms. Okay. So the use of restriction enzyme and ligase will allow us to make different combinations of your plasmid in vitro, that is under the test, in the test tube or in some uh, you know laboratory setup. So the product form is called as the recombinant. Okay. Now these things we have already spoken. So when this piece of DNA is transferred into the bacterium, okay, what it will do? It will use the host. So it will use the host DNA polymerase enzyme and it will undergo multiplication, replication. And this process of, or the ability to multiply its copies, to make more copies of that particular gene, that is we were talking about the antibiotic resistance gene, so it is called as the cloning, and in this particular case it is the cloning of the antibiotic resistance gene. All right, now let us see this particular picture. So what it has to do in this particular picture? Okay, so now here we have a piece of DNA. Okay, suppose it is coding for some particular enzyme or say some protein. So it is called as the gene of interest. Okay, now this is the gene of interest. Okay, now here we have the plasmid. Okay, now this plasmid and this, okay, this is already now cut. Okay, it is the required piece of DNA. Now here we have the plasmid. Now, here probably there were more parts of your DNA. Okay, so now this and this plasmid has to be cut with the same restriction enzyme. So we call it as digestion. Okay, this plasmid is obtained from the bacterial cell and the DNA and the plasmid, they have been cut with the same restriction enzyme, and this piece of DNA what we have is called as the gene of interest. Okay, it is called as the gene of interest. Now what is done is, we will have the, because it is cut with the same plasmid, we will have the same type of ends, okay, which are called as the sticky ends. Okay, you must have seen velcro. Okay, you must have seen this velcro, which is used for sealing your different things. Okay, all right, so this, what is the function of this velcro? Okay, they both are complementary, they will touch each other, right? All right, so same way the cuts produced here, they are also complementary to each other, they will join together. Now this joining is done by your ligase, DNA ligase. So this product of the combination of gene of interest and plasmid what we have, is called as the recombinant DNA. Okay, it is the combination of your different DNAs. Alright. So after this recombinant DNA, it is introduced into the host. So this host may be some bacterium, okay, for example E. coli, where okay it will go multiplication. So and it will produce the multiple copies, okay, multiple copies of this recombinant DNA. So when we break open, suppose if we have to isolate these pieces of DNA, see here you can see only one recombinant DNA, but here inside this progenies there are three. Okay, so if we want to isolate this, we'll have to break open this cell so that these plasmids are released out into the, okay, they are released into the environment. When I'm talking about the environment here, it is the, okay, it is the test tube. All right, so now let's move on to the next part. Okay, so here we will talk about the steps which are involved in modifying an organism. Okay, so what is done to modify an organism? Okay, so when I'm talking about an organism, basically I'm talking about the bacterium. Okay, now there are three different steps which are involved over here. The first one is the identification of the DNA. Okay, so we are not going to use any part of the DNA, because we have to look for the function. What is the end result that you are looking for? Okay, so we will look for the DNA, part of DNA which is having the gene. So let's call them gene of interest. Second part, what is done is the introducing this gene of interest into the host. Okay. Third part is maintenance of this new DNA into the host and then transfer of the DNA to its progeny. So basically these are the three main steps. First one is to identify the gene of interest. Secondly, to transfer it or introduce it into the host, and the third one is to maintain this into the host so that we get more number of progeny. So when we get more number of progeny, obviously there will be increased, okay, in the desired product. Okay, so now let us try to understand this figure here. Okay, so now here we have taken the human cell, okay, and here we have a bacterial cell. Now this human cell, okay, we have isolated a gene which is responsible for human growth hormone. Okay, so this pink piece of patch of DNA, okay, it is having the area which is coding, the gene which is coding for human growth hormone. Okay, and here we have the bacterial chromosome and the plasmid. So we have isolated the plasmid. But if you see here, this gene is also having, okay, the eco R1 site, okay, the restriction site or the enzyme, okay, site for this eco R1 enzyme, and your plasmid is also having the same site. That means both this, the piece of DNA of the gene of interest as well as the plasmid, can be cut with the help of eco R1 restriction enzyme. So when we do that, okay, when we cut, okay, there are this two pieces generated which are joined together with the help of your ligase enzyme, and the product that we have, the plasmid plus the gene of interest, is called as your recombinant DNA. Okay, now it will have the plasmid part as well as the uh, piece of gene which will code for your human growth hormone. So when this is introduced into the host organism, what will happen is it will use the host machinery to replicate this plasmid along with the piece of DNA. So on replication, there will be multiple copies of the plasmid present. So even when this bacterium is going to multiply, there will be an increase in the number of uh, plasmid carrying the gene of interest, that is the human growth hormone. So together, suppose see this is present in a test tube, okay. I'm very bad at drawing. So we will have multiple copies of this human growth hormone present over here. Okay. So when this gene is secreted, we will have more of the hormone. This is one example we have taken. Okay, so this is applicable for so many different sorts of proteins. Okay, we have seen what is biotechnology, what are its principles, okay, what is meant by restriction enzymes, what the process or the principle behind naming the or the nomenclature of your restriction enzymes. And till then, keep studying, keep revising, and keep smiling. All the best for your exams. Thank you.